Any announcements?
Yeah, I just got one.
All right.
So I just wanted to introduce Freddie, whose last name I don't know because I only know him as Freddie. So Freddie is a student of Sean Revito's, who many of you know, and he accompanied us out to the islands when we went out off Mexico early this year, and he's doing this cool project on Urosaurus. So he's going to be at the MBZ for the next 3 weeks doing library preps up in the lab, but make sure to chat to him. He's got a really fascinating project about island colonization, and if you look at these lizards, they're like a luminous liquid blue. So it's got some cool photos.
Anything else? All right, Rory's going to introduce today's speaker.
So it's my pleasure to introduce Phred Benham. Phred did his undergrad at LSU and then did a master's thesis on hummingbirds with Chris Witz, who spoke just a few weeks ago, and with Jim, who was an author on that paper, a really beautiful paper in Journal Biogeography. And then he moved to University of Illinois to start working in Zach Chevron's lab on the Savannah Sparrow system, and then subsequently moved with Zach when he moved to University of Montana, where he just finished up his PhD earlier this year. And then I was very fortunate that he decided to pick my lab to come. He has one of these NSF independent postdoctoral fellowships that tries to match sort of using museum-based collections in innovative ways. And I wish I had a lot of dirt on Phred, but I don't know him well enough that and his advisor didn't give me anything suitably embarrassing. So when he presents in his postdoc research, that'll be the opportunity. I know, but Libby was not going to be here. So, but apparently Davis is shut down completely because of the air. But anyway, not to get further distracted, it's really a pleasure to have him. He's sitting in the office on Michael and my side of the building, right in the back. His work is really interesting. It integrates many aspects that we're interested here in the MVZ, from looking at phenotypes of museum specimens, modern specimens, all the way to doing genomics and aspects of adaptation with these really fabulous sparrows. So he's going to be telling us about his PhD work and how that's extending into some of his postdoctoral work today. So thank you.
Yeah, thanks, Rory. Thank you all for coming today. It's been really exciting being part of the MVZ for the past few weeks, and I'm really excited to be here for the next 2 years. So today I'm going to talk a little bit about my dissertation work and hopefully explain a little bit of what my hopes are for the next couple of years while I'm here. The geographic variation is ubiquitous in nature. We see it across the tree of life in essentially every phenotypic trait that's been measured. And it reflects sort of dynamic interactions between a number of ecological and evolutionary processes, and thus provides abundant opportunities to understand the mechanisms underpinning biological diversification. And to me, one of the most striking patterns of geographic variation is the often strong association between phenotypic variation and spatially variable environments. And this includes some classic examples such as colored polymorphisms to maximize crypsis in different soil types, for example, in the rock pocket mice in the southwest United States, or between physiology and the abiotic environment, such as thermoregulatory performance in response to different temperature regimes in anolis lizards. So these phenotype environment associations are thought to evolve through a few different mechanisms. One of the primary mechanisms is via local adaptation, which leads to the evolution of higher fitness in the native habitat relative to genotypes from a foreign environment, and relatively lower fitness in a foreign environment relative to genotypes native to that environment. The second mechanism is through the evolution of phenotypic plasticity, where a single genotype can actually produce different adaptive phenotypes in response to different environmental cues associated with the different environments. And although we have a decent understanding of how these mechanisms might contribute to patterns of phenotype environment association, a number of outstanding questions remain, including how does population history interact with selective pressures to shape adaptive outcomes? Under what context does gene flow promote versus constrain adaptive divergence? And what role does plasticity play in adaptive divergence between environments? I think to make progress on these questions will require placing observed patterns of phenotypic variation within the context of how differential fitness evolves between these different environments. And this is differential fitness is thought to emerge from differences in organismal performance among individuals, which is in turn underpinned by variation in a hierarchically integrated suite of traits, with ultimately allelic variance underpinning trait variation, and then selection operating on differential fitness shaping allele frequency variation. Of course, I think this would be convenient if nature was this simple, but I think this is a gross oversimplification, and what might be more realistic perspective is that we have multiple metrics of organismal performance underlined by a whole large host of traits, from gene expression patterns all the way up to whole organ system function contributing to performance, and then a large number of potentially interacting loci contributing to trait variation. And I think taking sort of this complex mechanistic perspective will be essential for understanding the evolution of phenotype-environment associations within the functional context of the traits being studied, the genetic architecture underpinning trait divergence, and then the different ecological and demographic contexts in which adaptive trait divergence is proceeding. So in my dissertation, I attempted to sort of scratch the surface of this complexity. I wanted to take this mechanistic approach to address two primary questions related to adaptive divergence in sparrows, tidal marsh sparrows. First is, how do demographic and ecological forces interact to shape spatial patterns of local adaptation? And two, what role does ancestral plasticity play in adaptation to new environments? So the focus of my work has been on tidal marsh systems, which are these really fascinating dynamic habitats where you get regular freshwater inputs from inland watershed systems and saltwater inputs from daily tidal flooding. So the dynamic nature of this habitat really is selected for very specialized taxa that show rapid divergence from close relatives in a whole suite of behavioral, morphological, life history, and physiological traits, making these really exciting arenas to study patterns of adaptive divergence. And from the perspective of terrestrial organisms, one of the main challenges is this daily inundation of tidal seawater, which means that a lot of the available drinking water approaches or exceeds the salinity of seawater, which imposes a severe osmoregulatory challenge on most of these animals, given that most vertebrates must keep their internal osmolality about 300 milliosmoles per kilogram, and sort of deviations from this level can lead to cell shrinkage, impairs protein functioning, and ultimately leads to death if it becomes a chronic problem. So vertebrates, or organisms colonizing this environment from the terrestrial realm, must deal with the issue that their primary source of drinking water is over 3 times what they need to keep their internal osmolality out. And for a lot of terrestrial vertebrates that lack the specialized salt glands you see in a lot of marine animals, this appears to have been a significant challenge, and there's only a handful of taxa that have actually specialized on this habitat. And you see a couple Nerodia snake species, a local favorite, the salt marsh harvest mouse. And then if you consider the avian order Passerculus, which is over 5,000 species of birds, you really only have a handful of taxa that are considered specialists on this environment. And this is despite these habitats being surrounded by sort of relatively diverse songbird communities. And interestingly, the bulk of these songbirds appear to be three closely related genera of sparrows from one clade of New World sparrows. So to understand how these terrestrial vertebrates adapt to the challenges of this environment, my work's focused on the Savannah Sparrow. This is actually one of the most widespread North American bird species. It's found in a range of open habitats from tallgrass prairies out east, tundra habitat, and of course salt marshes along the Pacific coast. And some early experimental work showed that there were stark differences in salinity tolerance between different populations of the Savannah sparrow, with salt marsh adapted subspecies able to maintain body mass while drinking a range of different salinities, including up to or exceeding that of seawater, whereas interior adapted subspecies showed drastic reductions in body mass and experienced about 60% mortality when drinking water that was about half strength seawater. In the 60s and 70s, a whole host of subsequent physiological research went into trying to understand these evolved differences in tolerance. And differences in tolerance are generally expected to be affected through divergence in osmoregulatory capacity between populations, with osmoregulatory capacity essentially being the ability to maintain homeostasis in saltwater balance and maintain a constant plasma osmolality. So one of the first things they found with these experiments is that the salt marsh adapted populations actually reduce their drinking rate in response to increasing salinity, whereas interior populations increase drinking rate, which increases salt intake and exacerbates the osmoregulatory stress. They also have been shown to increase their ability to excrete salts into the urine, their ability to retain water in their system and maintain plasma osmolality at constant levels, and then underpinning increased water retention and salt excretion appears to be divergence in a number of kidney traits, including overall kidney size, the volume of the medulla, the length of the loop of Henle, total number of nephrons, and in particular the length of the loop of Henle and medullary volume appear to be important, as these are increased length of the loop of Henle and larger medulla is associated with being able to build up a steeper osmotic gradient and produce a more hyperosmotic urine and retain more water. Finally, another way birds could retain water is by reducing reliance on evaporative cooling when thermoregulating, but this hasn't been tested in tidal marsh birds. So despite a fairly large amount of research on the physiological mechanisms allowing these birds to persist in salt marsh, all of this work is focused on the Belding Savannah sparrow, which is conveniently located right outside of UCLA, where George Bartholomew was. But there's 6 other subspecies of Savannah sparrow that occur in tidal marshes. But it's unknown whether, including some that have morphological affinities more similar to these interior subspecies, so it's unknown whether there is variation in osmoregulatory trait divergence among these populations, and whether things like demographic variation might shape patterns of adaptive divergence in these different populations. So for the first part of my dissertation, I set out to address this question of whether demographic and ecological forces interact to shape patterns of osmoregulatory divergence in these tidal marsh populations. And it was mostly focused on these traits shown in blue. So, interpretation of phenotypic differentiation is generally confounded by a number of historical and demographic factors that interact with differential selective pressures. One approach to account for the influence of historical demography on would be to compare patterns of adaptive divergence across a number of populations of the same species exposed to similar environments within a phylogeographic context. And within this framework, you could examine how variation in gene flow among populations shapes patterns of divergence, with populations experiencing greater levels of gene flow between environments expected to exhibit reduced divergence. You can measure differences in effective population size, with selection predicted to operate more efficiently in larger populations. And finally, you could look at variation divergence time, with older populations predicted to have had more time to reach adaptive optima in novel environments. So first, so to begin, I start with my dissertation. I constructed a range-wide phylogeography for this Savannah Sparrow. I included 191 individuals, including 15 of the 18 described subspecies. I included 4 closely related sparrow species as outgroups. I sequenced DNA using a double digest GBAS-like approach and assembled breeds de novo in the stacks pipeline, just a standard pipeline. And analyses of the resulting SNP data set revealed 2 main clusters within Savannah sparrows. One is large blue polytomy, which spans most of the North American breeding distribution of the species, and it interestingly includes tidal marsh populations from the central California coast here in like the San Francisco Bay Area. And then a second cluster in green, which corresponds to the tidal marsh populations in northwest Mexico, sort of south of Santa Barbara. The next step was to visit these tidal marsh populations, and so I went to 5 tidal marsh localities in northwest Mexico, 5 along the central California coast, and then 3 interior freshwater populations in California. At each site I captured birds, measured urine and blood osmolality, measured total evaporative water loss using flow-through respirometry techniques, and collected whole kidneys to do a bunch of morphological analyses on the kidney tissue. And the results of this in situ physiological survey can be seen here, where along the x-axis you have the mean trait values for the 3 interior populations, and then deviations from that are effect sizes calculated based on t-tests between either the California tidal marsh in the interior or the Mexico tidal marsh in the interior. And so for kidney mass, medullary volume, plasma osmolality, and urine osmolality, you see Mexican birds consistently show greater divergence in these osmoregulatory traits relative to California tidal marsh birds. You see a similar trend in the urine to plasma osmolality ratio, although that wasn't quite significant. And then evaporative water loss, you actually see in California they show greater evaporative water loss, which is the exact opposite of what you would predict if you would expect tidal marsh birds to be reducing water loss relative to interior counterparts. So what explains variation in phenotype and physiological divergence in response to qualitatively similar environments? So I estimated a number of these demographic parameters using the program DADDY to compare 3 interior populations to each of the 10 tidal marsh populations I measured. And so we see significant variation in migration rate with Mexican populations showing relatively lower levels of migration rate from the interior compared to California. Effective population size was shown to be larger in California relative to Mexico. And then divergence time, although not significantly variable with latitude, tends to be greater in Mexico. I'll spare you the time. Those don't seem to be important in explaining trait variation. And so I'll mostly focus on this prediction that with increasing migration rate from the interior, you'd expect to see reduced trait divergence. Chris. Second compounding factor here could be the stark differences in other environmental parameters that vary along the Pacific coast. I tend to be a fairly lousy botanist, but I can notice some differences in vegetation between Northern California and Baja California. And so these differences reflect the general decline towards hotter and drier conditions in southern Baja California. relative to Northern California. And these hotter and drier conditions could exacerbate osmoregulatory stress and contribute to selection for increased osmoregulatory trait divergence in Mexico. So to simultaneously account for the influence of migration rate and variation in abiotic variables, I used a structural equation modeling approach to see how migration rate versus ecological divergence or differences would influence trait divergence for each of the 6 osmoregulatory traits that I measured, while accounting for covariance along that longitudinal gradient between the two. And in this model, ecological differences was a latent variable inferred from 4 abiotic parameters. One was osmolality measured from water samples taken at the site of capture for the birds. And then I took the average May-June precipitation, minimum temperature, and maximum temperature from the World Clean data set. May and June was when I was sampling these birds. So, and the results of these analyses can be seen here, where you have the different coefficients associated with each of the predictor variables shown on the y-axis. And for kidney mass and total evaporative water loss, you see a significant, that migration rate was the only significant predictor variable for variation in trait divergence for those 2 traits. Whereas for metrics of urine concentrating ability, urine osmolality, and urine to plasma osmolality ratio, it was actually these ecological differences you see among sites that were explaining variation in those trait differences. And then plasma osmolality actually seemed to be explained by both variation in migration rate and variation in ecological differences. And then medullary volume seems to be more strongly influenced by migration rate. That wasn't statistically significant. And I visualized some of these correlations using regression analyses. You can see the strong relationship between kidney mass and migration rate. And you do see a significant correlation between medullary volume and migration rate. And then max temperature turned out to be the most significant ecological variable, explaining variation in urine osmolality, urine to plasma osmolality, suggesting that these hotter temperatures experienced in the Mexican populations is selecting for the ability to excrete greater amounts of salts in the urine. So to bring all that together, Mexican tidal marsh populations exhibit larger magnitude of divergence in all of these osmoregulatory traits. And variation in migration rate among populations seem to explain this greater divergence in Mexican populations for evaporative water loss and variation in kidney morphology, whereas maximum temperature seemed to explain the variation in salt excretion ability. Interestingly, this higher level phenotype seemed to be explained by both of these metrics. All right. So based on the results, we concluded that demographic and ecological parameters can differentially influence interacting traits that contribute to increased osmoregulatory capacity. And this underscores the importance of simultaneously quantifying ecological and population genetic parameters to elucidate the mechanisms driving patterns of geographic variation. So for the second part of my dissertation, I want to focus on what role does ancestral plasticity play in adaptation to these tidal marsh environments. And really the motivation for this part of my talk, or for this part of my dissertation, comes from the fact that most of the close relatives to Savannah sparrows are freshwater adapted, and Savannah sparrows themselves are largely this unresolved polytomy at the base with a cluster associated with Mexican salt marsh populations and these California salt marsh populations, suggesting that tidal marsh Savannah Sparrow is likely to derive from a freshwater adaptive ancestor. However, we also know that exposure to salinity in these freshwater adaptive populations can have severe consequences for these birds, begging the question of how they manage to colonize these habitats in the first place. So adaptive plastic responses represent one potential mechanism allowing these birds to get a toehold in these new environments. And some of these plastic responses can be seen on this bullseye figure, where the center represents the adaptive peak in the novel environment. And you might expect some cases where adaptive plastic response puts the population right at the new adaptive peak in the new environment, although this is generally considered to be pretty unlikely. But what might be a more likely response is where you have a variable adaptive plastic response that puts the population within the realm of the adaptive peak, and then subsequent selection will move it towards the adaptive peak. This has been shown in Daphnia, Sierra Nevada Daphnia, where UV exposure had traditionally selected for more melanic individuals, but with the introduction of fish predators, you saw a shift towards lighter colored individuals being favored, shown by that black triangle on that fitness landscape. And Scoville and Frender found that within an ancestral-like population, expression in the ebony and DDC loci moved the population towards the adaptive peak in the new towards the adaptive peak, but not all the way there, showing that, yeah, it's shown by the white circle. Genetic accommodation is thought to be the main process by which an ancestral plastic response places a population within an adaptive peak and allows for selection to then drive the population the rest of the way. In this process, exposure to a novel environmental pressure unmasks a large amount of genetic variation, resulting in these large amount of phenotypic variation in response to the novel pressure, with these individual differences in phenotypic variation referred to as reaction norms. And then with continued exposure to the same novel environmental pressure, you'll expect canalization of this reaction norm variation, which can lead to the movement of the mean trait value in the novel environment up and elevation of the overall slope over time. Theory has demonstrated that this process likely occurs, and there's a few good lab experiments that also demonstrates that this process can occur, but there's sort of still very few compelling examples from natural systems. And to document the process of genetic accommodation occurring in natural systems requires comparative analyses of acclimation responses to similar environmental pressures in both an ancestral and derived population to test a number of predictions consistent with genetic accommodation. The first would be that ancestral acclimation responses are going to be in the adaptive direction. And second, selection on plastic responses in the derived population will lead to increased trait elevation and slope, or slope. And then finally, given that you have these ancestrally variable reaction norms, you expect selection to operate on this variation to reduce variance in environmentally induced trait means. So to test this first prediction, I brought 43 birds into captivity from population in western Wyoming, which is a freshwater adapted population. I acclimated them to captivity for 36 days. Afterwards, I divided them into two groups, one which was just maintained on freshwater and served as a control, and then the other group which I gave a sort of mildly saline drinking solution to and sampled out 1 day, 7 days, 15 days, and 30 days of salt exposure to get a sort of understanding of the time course of acclimation response in these populations. And so as I mentioned earlier, these birds are going to need to maintain their plasma osmolality at a constant level to avoid the negative consequences associated with deviations from their normal level. And they're predicted to do this either through behavioral responses such as reducing salt intake, mechanisms that increase salt excretion or water retention, which will ultimately reflect changes in kidney morphology. So with looking at plasma osmolality, we see that there's no significant differences between treatments or across sampling times, suggesting that they are, in face of these salt challenges, able to maintain a constant plasma osmolality level. Looking at changes in drinking rate over the course of the experiment, you see that in birds maintained in freshwater, there was absolutely no change in drinking rate, but you see a significant decrease in drinking rate over the course of the experiment in the salt-exposed birds, suggesting they are behaviorally reducing salt intake to cope with. You also see evidence for greater water retention as measured by just increased relative plasma volume, which they appear to increase plasma volume within the first 24 hours and then maintain it at higher levels throughout the course of the experiment. And then to get a comparable measure of salt excretion between both the freshwater exposed birds and salt exposed birds, I gave all birds a dose of 0.6 molar sodium chloride water, so that I wasn't just measuring diet differences between the two groups. And then I collected urine samples every 20 minutes afterwards for 2 hours. And then I took 2 metrics of urine concentrating ability. The first is just sort of the peak osmolality an individual bird was able to reach, which is referred to as Umax. And then the second one is the sampling time at which they reached Umax, and with lower U times expected to be associated with a greater efficiency of getting rid of salts from the system. These are data just from the 15-day sampling point. You see there's no difference in Umax between the 2 treatments, but you do see a significant reduction in U time in the salt-exposed birds, suggesting they are more efficient in clearing salt loads from their system than freshwater exposed birds. So there's some evidence that salt excretion ability is plastic there as well. So looking at kidney morphology, just kidney mass, you see the kidney mass increases through most of the course of the experiment. And then looking at more relevant kidney morphology trait, medullary volume, you see that after 7 days you have a significant increase in kidney or medullary volume, and that remains larger throughout the course of the experiment. So yes, we should have some evidence that ancestral acclimation responses are in the direction expected for tidal marsh divergence. So to test the second prediction, I've replicated the same experiment with 14 birds captured from the California tidal marsh. And I only sampled these the 15-day period after which we had seen most of the divergence between between treatments in Wyoming. Although again, we see no difference between treatments in plasma osmolality. We see again that these birds are tolerating a higher plasma osmolality relative to interior populations. They show the same decrease in drinking rate. The freshwater exposed birds also showed a significant decrease in drinking rate, which I have not found a good explanation for why they did that. You see see higher water retention in salt-exposed birds and almost significant evidence for increased plasticity in water retention in the salt-exposed birds from California. Looking at our two metrics of urine concentrating ability, there's a significantly greater urine concentrating ability in the tidal marsh populations, but no difference between treatments. But you see sort of comparable levels of plasticity and U time between treatments in both populations. And then finally looking at these metrics of kidney morphology, you see plasticity in medullary volume, but an elevation of the overall reaction norm of medullary volume, and there's actually no difference in overall kidney mass in the tidal marsh population. So in at least some of the traits, we see evidence for this second prediction, that there's been change in the overall reaction norm in the derived population. So finally, to test this, tested this last prediction that you'd expect to see reduced variance in environmentally induced trait means. And sort of to illustrate what this means, I plotted individual level reaction norms of drinking rate for all birds. And the birds sampled at 15 days are colored and all the other ones have been grayed out. And within this first 24 hours, you see a tremendous amount of variation in the response to drinking salt water, with some birds increasing and some birds decreasing their drinking rate. And if you contrast that with the responses in the tidal marsh population, you see a more consistent decrease in drinking rate. And to quantify that variation, I used the coefficient variance metric. And you can see that that initial change in drinking rate, there's significantly less variation in the derived tidal marsh population relative to the ancestral population. And looking at a number of other traits in the salt-exposed birds from Wyoming and California, you can see the reduced variation in plasma osmolality, plasma volume, and U time in the California population, but actually the opposite pattern, where you see increased kidney mass, increased variance in kidney mass, medullary volume, and slightly more variance in Umax for the California population relative to the Wyoming population. A number of other processes could also reduce variance in trait means, or reduce variance in these traits, such as bottlenecks associated with colonization of tidal marshes. But if something like a bottleneck were explaining these patterns, I expect it to be true across the board. And I also measured a trait which is not expected to be under differential selection between populations, tarsi length, and there's no difference in variance between population and that metric, suggesting that differences in reduced variance in some of these traits in the tidal marsh population is likely due to selection. So we find some support, again, for the third prediction. If you put it all together, you see fairly idiosyncratic support for this genetic accommodation hypothesis across the different traits, with actually the ability to retain more water as measured by plasma volume being the one trait that stands out as showing the most support for genetic accommodation process. All right. And so to summarize all of this, I think by analyzing patterns of geographic variation within this mechanistic framework, I hope to have shown that patterns of non-parallel adapted divergence in tidal marshes reflect variation in both immigration rate and ecological differences among these different populations. The ancestral plasticity appears to play an important role in adaptation to the tidal marshes, but only in some of these traits. And I think together these results are highlight that different mechanisms can shape patterns of geographic variation in different traits that together contribute to organismal performance, requiring really a careful evaluation of the different traits involved in contributing to tolerance and fitness, and how a variety of different evolutionary and ecological mechanisms might shape divergence in these complex integrated systems. All right, and yeah, looks like I still have a few minutes. So I kind of want to just segue into talking about what I plan on doing for the next couple years here. And a lot of the motivations for my postdoc were sort of from the realization that we may not be sort of capturing what the geographic variation may have actually been like 100 years ago, especially if you're considering places like the California coast, which have seen a dramatic amount of alteration over the past 100 years. And looking at places like the San Francisco Bay Area, where sort of between the 1890s to 1930s, there was a dramatic amount of what they called evaporative salt mining, where they basically diked off and converted tidal marsh habitat to these evaporating ponds to produce salt, which is thought to account for about 45% of the destruction of tidal marshes in the San Francisco Bay Area. And then also in the area, we well-known urbanization and human population growth, which accelerated after World War II, together with tidal marsh development, contributed to over 90% habitat loss in the Bay Area between 1900 and the present. So then on top of this widespread habitat destruction, you also have increasing salinity over the past 100 years, and of course increasing temperatures through time as well. So in light of all these changes, I wanted to see whether you have temporal and spatial variation in locally adapted loci, where I would predict that you would have variation in allele frequency in response to some environmental parameters such as maximum temperature and space, associated with variation in the same environmental parameter through time. And the sort of second objective would be to relate these historical events that contributed to tidal marsh destruction with demographic changes in Savannah sparrow populations, with prediction would be that with the destruction of tidal marsh populations, or tidal marsh habitat, you'd see reductions in effective population size and changes in connectivity and gene flow among populations. One prediction could be that you see increased immigration rate into these tidal marsh populations as they become a sink population. And then finally trying to tie these selection and demographic changes to phenotypic variation and adapted in a functionally meaningful trait, which would be bill size. Bill size has been shown to increase in many species of tidal marsh sparrows, which is in a way associated with that evaporative water loss. Sparrows with larger bills can dissipate more body heat through the bill, which reduces reliance on evaporative cooling. It's thought to be a mechanism to conserve water in water-limited environments such as tidal marshes. So to address these questions, I'm planning on taking advantage of the great temporal and spatial sampling of Savannah sparrows in the museum collections. And I hope to take an exome capture approach to sequence individuals from across the latitudinal gradient. So most of the specimens I collected through my PhD, and then replicate that same latitudinal transect prior to 1920 to look at changes in allele frequency along that gradient, but also migratory connectivity among populations, among different tidal marsh populations, and within the interior and salt marshes. And then I plan on taking a more focused temporal sampling from the San Francisco Bay and sampling 10 sparrows per decade between 1890 and 1930. Then there's a museum gap, and then having a sixth population, which is basically from my dissertation, to see how, get a better time series of how these things are responding to different selection pressures and to habitat destruction. And bill size is something I've already been working on here. And looking at this metric bill surface area, which is basically the area of a cone, it includes bill width, bill depth, and bill length, which we would predict to get larger in populations experiencing greater temperatures or salinities. We see a trend towards larger bill surface area in modern specimens compared to ones collected before 1921. That wasn't quite significant, but interestingly, if you break up the components of bill surface area, you actually see an significant increase in bill length between the historic set of specimens and the modern, but decrease in bill width over the same time period. So suggests that birds are overall getting narrower and longer bills. These are data I just analyzed a couple days ago, so we'll see how this plays out. All right, and with that, I'd like to acknowledge my old lab, the Chevron lab, all committee members and wonderful undergrad assistants, all my funding sources, and of course the huge number of museums that contributed to shoes and things like that.
We have time for questions. If somebody can get the lights, please.
Yeah.
So, with these salt marsh specialists, are there any behavioral differences that could also be an accommodation for the salinities, such as maybe trying to take in water from, lower salinity sources versus high salinity sources?
Yeah, I mean, it's one thing that's been really hard to document in the field. There's some thought that they might be able to select for dew. But there's a lot of variation in the plant species in these tidal marshes, with some getting rid of their salt into the dew, whereas others don't do that. So it's been hypothesized that they may be choosing between plant dew. In some of these places, the freshwater sources are extremely limited. If you get down into Baja California, there is no water outside these estuary systems. It's really dry. So it's, yeah, the other possibility is dietary inputs, but they're also eating osmoconforming mollusks and things like that, so they seem to be able to do fine. But yeah, there's just very limited amount of behavioral work that's been done.
And then secondly, I noticed on your map that there was that break seemed to be right around the transverse range, where you had that southern population, south of the transverse range, and then the, widespread population north of the transverse range.
Yeah, that's the one that cuts across the Southern Valley. Sorry, I'm still new to California. So yeah, there's a lot of phylogeographic patterns that I kind of glossed over. Historically the break was thought to be at Point Conception because it's all coastal in California, and then everything north of there was thought to be sort of the nominate subspecies group, and everything south, Belding Eye group. So my work has shown that Morro Bay actually appears to be a contact zone between the two groups, and there appear to be completely admixed at Morro Bay. The extent of that in either direction is still sort of an outstanding question, but yeah, there does seem to be a meeting point right around there. Santa Barbara area. Yeah.
Do you think that the change in bowl could be driven by a change in diet? And I'm just thinking if they've switched more to insects, they would select for a longer, narrower.
Yeah.
wonder if you could use isotopes to
Maybe.
get at that.
Yeah, so, yeah, if it was switched from a primarily carnivorous to primarily insect. Yeah, that's the only reason I would expect a narrowing of the bill, because you'd expect overall surface area to increase for thermoregulatory performance. Yeah, so like I kind of had alternate predictions for how bill morphology could change. One could be if you were getting increased immigration pressure, you may have this working against divergence in response to changing temperature or salinity. So, yeah, we'll see if I get into isotope analysis. Yeah.
There's also the introduction and proliferation of like fennel, and isn't that really easy for birds to feed on? Right?
Yeah, they definitely seen a lot of them in fennel.
Yeah.
So, yeah, it could be various introduced species.
I was wondering in a broader sense if there's something about sparrow biology in particular that enables them to cross different groups of sparrows on the east and west coast to
Yeah.
sea
Sparrows
land.
seem really good at getting into tidal marshes, and it doesn't seem to be necessarily association with open habitats, because there's plenty of genera of sparrows in open habitats that haven't gotten into tidal marshes. As far as whether they're more plastic in these traits, there's rufous-collared sparrows show some of the same sort of plasticity patterns in these osmoregulatory traits, and rufous-collared sparrows are related to white crown, or like the South American equivalent of white crown sparrows. I mean, some of the work I've done with Jen Walsh and others has shown that there's actually not a lot of parallel genomic divergence in a number of these sparrow populations. There's a few genes there, like 2 or 3 of the sparrow species show divergence between upland and interior populations. So it's not. It also doesn't seem like there's like some genomic variant at the base of the sparrow tree, or the base of that clay that's allowed them to colonize. So I don't actually know what's going on with sparrows and why they seem to be particularly good at this.
I got a question about the Baja populations. How long have they been there? And it looked like it was a much older divergence than the rest of them across North America.
Yeah.
And the question is, what was the habitat like during the time when they colonized, and might there have been intermediate habitats that would have facilitated?
Yeah, so for the phylogeographic analyses, I estimated it to be about 480,000 years ago, but quite a large range on the other side of that. I think that's within when we know that these habitats, salt marsh habitats have formed. They seem to have, more work has been done on habitats like around here, where they thought to have formed over the course of the Pleistocene. The Gulf of California is thought to have begun forming 5 million years ago, I think. So I'm here to stun more phylogeography than me. But yeah, I think these tidal marsh environments have been there for at least that time scale.
Jim, you look like you were gonna.
The tidal marsh would have been there for a long time, but terrestrial freshwater ecosystems would have been there throughout the Pleistocene, or at least periodically throughout the Pleistocene. So you can't look at Baja as it is today, going back 400,000 years.
Climatically, yeah.
Climatically.
So like the forest would descend out of the mountains.
Well, yeah, we know from the fossil record, from the Midden record, for example, that the coastal woodrats that are here in Southern California went all the way down to as far as central Baja. 50,000 years ago, within the last 50,000 years.
Well, yeah, I was just thinking, if you have some plastic responses that are not adaptive, like drinking more if you end up in a real salty area, but there's a habitat that's somehow intermediate, so if you have a source of fresh water, that might facilitate colonization incrementally of a salt marsh.
Yeah, and it does seem like recent colonists on the East Coast do tend to be in more brackish habitats. So there's like a swamp sparrow subspecies and Nelson sparrow that tend to occupy more brackish habitat than the really older divergences. But yeah, it could have facilitated some of this.
Other questions? When
So what do you measure evaporative cooling? That's just through panting, right? Or how often there?
So I use a flow-through respirometry system. So basically you, it's a fairly complex system. You're pumping dry air through a thermal cabinet, which keeps the birds at a constant temperature within their thermal neutral zone, so they're not, increasing metabolic rate to thermoregulate in any way. And then you have a control chamber and an animal chamber, and then you basically measure the difference between those two chambers in, relative humidity or total water pressure, or one of those metrics. And so then you get total evaporative water loss, which is cutaneous and respiratory, which would be the panting. And so, yeah, once you get it all set up, it's fairly straightforward to analyze.
Okay, let's thank Phred one more time.